Hello friends, welcome back to my YouTube channel Nasrin Padasale. So in this video, I'm going to explaining you important topic from which question has been asked for this joint CSIR UGC NET June 2022 examination. So I already uploaded the shift 1 question paper friends. So if you didn't watch the important topic from which question has been asked for shift 1, please watch the video that I'll be giving you the link of that video in the description below. So first with regarding to part B, I am going to discuss. Question has been asked from diuretics like reabsorption of water and NaCl by kidney. Next, program cell death during tetrapod limb development and shell and weight of mollusks that is attacked by predator. Next, a question asked from extinct animal and endangered animal, and they are asking the reason for the extinct animal. Okay. Next, they were asking question from the DNA replication both in prokaryotes and eukaryotes. Next, alpha, beta, gamma diversity, and there were questions regarding to sex determination pathway and plasma membrane of plant, animal, bacterial, mammal, uniqueness about each of this particular thing. Next, what is the catabolic end product of urine, that is from de novo nucleotide synthesis? This question has been asked, and what they are asked, what is mean by pericide, and their question with regarding to self-surface labeling technique and HPV vaccine or how it is made, like that they were question asked, and ecotones, GA, gibberellic acid, and protein domains question has been asked, and AFLP technique question has been asked, and bioaugmentation, bioremediation question has been asked, and with regarding to savannas in Indian bio subcontinent question has been asked, that is from woodland grassland ecosystem question has been asked, and from regeneration, that is in developmental biology regeneration question has been asked, and question has been asked from hemoglobin present in diabetic patient and the uniqueness about that particular hemoglobin, and the question has been asked from RNK selection, and question has been asked from this post translation modification, that is specifically histone post translation modification associated with the DNA repair mechanism, and they were asking a species, that is a name of bird which are known to different flowers, and question has been asked with regarding to methods that are used for selectively lysing a newly dividing cell, like which chemical is used. Okay. Next, a question has been asked from statistical side, biostatistics. Few question has been asked for shift 2. That is what is mean by sensitivity and what is mean by specificity. If you know the meaning of both, you can easily answer those questions. And there were question asked from protein structure and folding and the centromere protein histone variant and chromosomal disorder. So whenever you are going to study chromosomal disorder, in addition to studying totally from textbook, go for referring few important and newly advanced that has been done in the chromosomal disorders. Okay, so you can refer this CDC website or few other international website. Okay, so from that particular website only question has been asked. Next, they were asking the action of photosynthesis inhibitor or inhibitor of chlorophyll electron transport chain. Next, they were asking question with regarding to Sherwood cap from ecology, cancer genes and their function and correlation coefficient from biostatistics, and potency, equivalence, competence, specification. What is speciation or specification? What is mean by that? And the question has been asked how to analyze the cell cycle progression for culturing mammalian cell. And question has been asked from dorsoventral polarity in Drosophila, extracellular matrix, dehydrates. Next, with regarding to parts, there a question has been asked from in vitro translation system, how it has been done. Special focus to SGSP. They had given a page gel and they'll be asking which of the following option is correct. Then there a question has been asked with regarding to phytochromes in plant, mitochondria, iron regulator element, Calvin Benson cycle. The question has been asked, and competition like intraspecific and interspecific competition, and they will ask for vesicle formation, transposition, retrotransposition, and Michaelis-Menten's equation. So same question has been asked, that is Michaelis-Menten equation has been asked in shift 1 also. Next question has been asked from thermodynamics like Gibbs-Free energy, thermokinetics, and resting membrane potential, treatment of cancers. Like there are questions regarding to when you treat an cancer cell with a drug compound X, and they are asking, they have given immunoblotting and they with a different type of specific marker, and they will be asking you to figure out whether the drug is efficient or not. Next, the question has been asked from segregation of chromosome, Indian biosphere reserve. So whenever you are studying, have ion biodiversity hotspot present in India, Indian biodiversity reserve, and what is the key fauna present in that particular reserve. And the question has been asked from plasma membrane protein, like plasma protein and blood clotting factor, MRI, Early spot, ELISA. And from black copper and marrow-diploid question has been asked, and homeostasis of iron, autophagy during embryogenesis and postnatal development. And the question has been asked with regarding to choral development. Next, transplantation experiment done in mouse. So that's why I'm asking you to, while studying transplantation and all, either go for QB book or go for referring few particular article, and cancer, like they were asking a description of cancer, and they were asking a match the following type of question, and insulin, they were asking a structure of insulin, and they had given a pure statement, and they were asking which of the following statement is either correct or incorrect. Next, amino acid biosynthetic pathway, anterior posterior body plan in Drosophila, and they were asking mechanism of submerged tolerant plant, Ramachandran plot, cladogram. And regarding to biostats, many question has been asked, like they are asking you to explain about ANOVA, Poisson distribution, standard error, kurtosis. So you at least know the definition of this biostatistical technique, even though if you don't have time to study about in-depth, or if you find any difficulty in going for in-depth analysis, just know for what purpose that particular technique is employed. Next, they were asking a signaling pathway specifically in development, like Wnt signaling pathway question has been came. Next, I have a question regarding to when you knock out APC cell line or macrophage cell line, and what is the possible consequence in T cell activation, like that. So only if you know the entire mechanism of T cell activation, you can able to answer those questions. Okay, so if you're studying about T cell activation, ultimately you should try to apply the concept in the question format. Okay, since the CSIR is a highly tough subject, you only if you apply your concept that you studied, you can able to clear this examination. Next, they were asking questions regarding to DNA replication, eukaryotic functions of different acting binding proteins, cell junction function, pedigree. Again, pedigree from 4 question has been asked for both in shift 1 and shift 2. Next, vulval formation is C. elegans, plant nodulation protein and the domain, and HFR mapping, ICU and red data book question has been asked, and blue white screen like alpha beta complementation, and from bioremediation question has been asked. And in addition to that, fate of mammalian genital ridge, like if you study the sex determination pathway itself, you can able to answer that particular question. And from gibberellic acid also question has been asked, and regarding to genome editing tool, like zinc finger nucleases, meganucleases, CRISPR-Cas system, talents question has been asked. So these are the list of topics from which question has been asked for shift 2 student. So I hope this video will be really helpful. If it is really helpful, then please click the subscription button. Thank you friends. Thanks for watching this video.